my channel, myself Dr. Surabhi Sahney. Today I am going to talk about part 2 of my video regarding important questions of microbiology for your theory exam. As you know, in my previous video of microbiology, I have covered the important questions of Unit 1 of microbiology. Here I'll cover the rest of the unit for your theory exam point of view. Begin with the second unit, that is immunology. The one of the most important question is of 5 marks, which is in the form of differentiate between active immunity and passive immunity. Next important chapter is immunoglobulins. So a 5 marks question may come on what is immunoglobulins, their types, their uses, their function. 5 marks question may come on secretory antibody. Along with that, a short note may come from this chapter on the topic IgA antibody. Begin with another chapter in this unit, that is the antigen antibody reaction. A long question may come on different types of antigen antibody reaction. And a short note may come on what is agglutination test, and another short note may come on what is agglutination reaction. Along with that, the most repeated question of this unit of microbiology is short note on ELISA. Either a 3 marks question or a 5 marks question may definitely come, as this question has been repeated more than 5 times in the last 10 years of microbiology. Viva based question is there, that is what is immunoelectrophoresis or prozone phenomena. So these 2 questions are the Viva based questions from this unit. Next important chapter is hypersensitivity chapter. I think this chapter is most important among this unit, because lots of long question may come from this chapter. First long question may come, what is hypersensitivity? A 5 marks question may come. Type 4 hypersensitivity, another 5 marks question may come. Type 1 hypersensitivity, another 5 marks question, or a question may come in the form of differentiate between type 1 and type 2 hypersensitivity. Another 5 marks question may come from this chapter. So this chapter of hypersensitivity is very important for your theory exam. Other short note may come on what is Arthur's reaction and what is serum sickness. These are the other 2 short notes that may come if they want to make your paper little tricky and hard. Begin with the third unit, that is bacteriology. Lots of queries have come regarding bacteriology, like ma'am, please tell how to learn bacteria, which bacteria is important, and lots of queries are there regarding bacteriology. So here I come with a small tip and trick regarding how to learn bacteriology. It's very easy. Learn a bacteria. Need to learn under 4 headings. The first heading is morphology of the bacteria. जब भी कोई बैक्टीरिया आएगा उसकी मॉर्फोलॉजी क्या है वो आपको लर्न करनी होगी. क्वेश्चन में पूछा हो या ना पूछा हो, आपको उसकी मॉर्फोलॉजी लिखनी है कि वो पर्टिकुलर बैक्टीरिया ग्राम पॉजिटिव है कि ग्राम नेगेटिव है, कोकस है या बैसिलस है, स्पोरिंग फॉर्म में है या नॉन स्पोरस है नेचर में. सो उसकी मॉर्फोलॉजी क्या है वो बहुत इंपॉर्टेंट होती है. सेकेंड थिंग इज याद करने के लिए है, वो ये है कि उसके अंदर कोई तो स्पेशलिटी होगी, लाइक कुछ एंजाइम होंगे स्पेशल्स, कुछ टॉक्सिन्स होंगे, तो उनके नाम आपको मेंशन करना जरूरी है कि एक पर्टिकुलर बैक्टीरिया या एक पर्टिकुलर बैक्टीरिया में ये टॉक्सिन है, ये एंजाइम है. तो जस्ट लाइक दैट यू नीड टू लर्न एंड राइट इन योर आंसर. द थर्ड हेडिंग एंड द थर्ड ट्रिक इज always and always write the pathogenesis of a particular bacteria, चाहे क्वेश्चन में पूछा हो या ना पूछा हो, क्योंकि अगर आपको यही नहीं पता होगा कि वो बैक्टीरिया मल्टीप्लाई कैसे करता है, किस एनवायरनमेंट में करता है, बॉडी के किस पार्ट में जाके वो मल्टीप्लाई करता है, हमारी जब आपको उसकी पैथोजेनेसिस ही नहीं पता होगी तो आप उसकी ट्रीटमेंट कैसे करेंगे? सो द पैथोजेनेसिस इज वेरी वेरी इंपॉर्टेंट. And when you know the pathogenesis, here come the fourth part to learn bacteria, that is the lab diagnosis. Even you can see a separate question may come on a lab diagnosis of a particular bacteria. Otherwise, if a 10 marks question may come either on a separately on a lab diagnosis, you have to write it, or if a direct question may come like you have to write in detail about the staphylococcus or streptococcus, then also you need to add the lab diagnosis part in your answer. In that you have to tell कि specimen कहाँ से उसके लेंगे, हम उसको कल्चर कैसे करेंगे, उसमें स्टेनिंग कौन सी होगी और उसका पूरा स्टेनिंग प्रोसीजर क्या है वो आप एक्सप्लेन करेंगे, उसमें आपने क्या क्या कंट्रोल लिए हैं, फिर आप उसमें एंटीबायोटिक टेस्ट का भी डिस्क्रिप्शन बताएंगे. एंड दिस एंटीबायोटिक टेस्ट डिस्क्रिप्शन इज वेरी वेरी इंपॉर्टेंट इन लैब डायग्नोसिस, क्योंकि अगर आपको यही नहीं पता होगा कि कौन सी एंटीबायोटिक इस बैक्टीरिया से कॉज होने वाली इन्फेक्शन पे वर्क कर रही है, how you will recommend it in the treatment of an infection which is caused by that particular bacteria. So that's why it is very very important to know about the antibiotic test in the lab diagnosis of all the important bacteria. And the last part that you need to learn and write in your answer is the treatment of that particular bacteria which caused that particular infection. So that's all about the tip and trick regarding in which headings you will learn the bacteria in microbiology. Example, a direct question of 10 marks may come on certain bacteria like Staphylococcus, 10 marks question, Streptococcus, 10 marks question, lab diagnosis on pyogenic meningitis, 7 marks question, a differentiate between Streptococcus viridans and Streptococcus pneumoniae, a 5 marks question, Mycobacterium tuberculosis, 10 marks question, a short note may come on miliary tuberculosis, that is the 5 mark short note, Widal test, a 5 mark short note, Corynebacterium diphthery, a 10 marks question. The question of the differentiate between actinomyces and nocardia, a differentiate between question may come, but it rarely come in your paper, but it's still very important. Salmonella typhi, that means the lab diagnosis of a enteric fever, a separate question of 7 marks is there, that is also important, or even a 10 marks question is there on this particular bacteria. So that's all about your bacteriology. Begin with the next unit, that is virology. I think virology is quite interesting and an easy unit of microbiology. Here in the first chapter you have to just read the general properties of a virus. Begin with the most important chapter in virology, that is the hepatitis, because of the questions may come on hepatitis. Just like a question may come on hepatitis B, 5 marks. Question may come of hepatitis A, 5 marks. Question may come on what is the antigenic structure of hepatitis B, that is also a 5 marks question. The fourth important question in hepatitis is the short note on hepatitis B vaccine, another 5 marks question. Even a question may come as a differentiate between hepatitis A and hepatitis B, another 5 marks question. That a short note may come on interferon, a 3 marks question is in that chapter. The important part is AIDS. So what is AIDS? A short note 3 marks question may come, or even a direct long question may come on AIDS as what is the transmission of AIDS and the lab diagnosis of AIDS as a 8 marks question. Along with that, the morphological structure of HIV is very very important along with its diagram. So that's all about the virology part in microbiology. So here I cover the 3 main units which are very important. One is the Immunology, second one is the bacteriology, and third one is the virology, along with the tip and tricks regarding how to learn bacteriology. So that's all about my video. Thank you for watching.